All right, do you guys see my slides?
Yes.
Okay.
We see your slides and we hear you.
Great. All right, so I'm Allison Leaf. I'm a scientific program manager at Seven Bridges for the NHLBI BioData Catalyst project. And today I'm going to share one of our primary use cases, which is multiple variant GWAS. This presentation is going to have two parts. I'll start off by sharing some background on the BioData Catalyst ecosystem and the multiple variant GWAS use case. And then in the second part, I'll talk about the tools and features that Seven Bridges developed for researchers performing this type of analysis. So as you heard yesterday, BioData Catalyst is a cloud-based data analysis system sponsored by the National Heart, Lung, and Blood Institute. And it's going to serve as the place for heart, lung, blood, and sleep researchers to find, access, and analyze NHLBI-funded omics and imaging data sets in the cloud. The system currently hosts data sets from the Transomics for Precision Medicine program, or TOPMed, and I'll say a little bit more about that in a bit. But first, I wanted to touch on the fact that BioData Catalyst is not just one platform, as many of you know. Instead, it's an ecosystem of cloud-based platforms and services that are integrated with each other to provide maximum functionality to the end user. So for simplicity's sake, and I know this is a wildly complicated diagram, you can think about the system in terms of a data layer, a search layer, and an analysis layer. So in the data layer, the NHLBI is hosting the TOPMed data in a cloud bucket for all of BioData Catalyst. So all the platforms and services in BioData Catalyst let users access this one copy of the data. And then UChicago is operating their auth service for dealing with user permissions on the data. And then in the search layer, Picture API, which we heard from yesterday, and the UChicago Windmill service let users query the phenotype data of the TOPMed studies, and then eventually the additional hosted studies on the system. And then finally, in the analysis layer, we have Seven Bridges and Terra providing workspaces for computing on genomics data at scale. As Manisha mentioned yesterday, the Seven Bridges BioData Catalyst platform is a separate installation than our other platforms. So it has the same front end and back end services as the Cancer Genomics Cloud and Cavatica platforms. The main differences really lie in the hosted data sets and the tools and features for analyzing those hosted data sets. BioData Catalyst currently hosts about 40 studies from the TOPMed initiative, but TOPMed actually has 80 different studies in total with subjects who have disorders of the heart, lung, blood, or sleep. And many of these studies have thousands of clinical variables that were measured, and there's whole genome sequencing data from about 150,000 subjects to date. Therefore, a primary use case for researchers studying these data sets is genome-wide association studies. So the researchers are taking a phenotype of interest and they're looking to find variants in the population that are significantly associated with the presence of that phenotype. So Seven Bridges has developed some new tools and features to enable researchers to perform GWAS on the system. It's important to note that about 97% of TOPMed variants are rare, and they occur at a frequency of less than 1% in the population. This is in contrast to the common variants that occur at a frequency of greater than 1%. In addition, many of these rare variants occur in only one subject out of the 150,000. So that makes the chances of detecting these variants as significant in a GWAS nearly impossible. So in order to study these rare variants, researchers are performing a specific type of association test called a multiple variant GWAS. And this is in contrast to a single variant GWAS, which can identify common variants that are significantly associated with a phenotype of interest. So the main point of this slide is that multiple variant GWAS lets users detect associations with the 97% of TOPMed variants that are rare. So this list shows the necessary components for performing an association test. And the first three are needed for both a single variant GWAS and a multiple variant GWAS. The last one is specific to the multiple variant GWAS. So first, researchers need a phenotype of interest, like high cholesterol. This is what they want to find variants associated with. Second, they need their VCF data for subjects with that phenotype of interest. Third, they need their association pipelines, and there are several different flavors of this out there. And then finally, if they're doing that multiple variant GWAS, they need a mechanism for creating variant groupings to increase statistical power when they're looking for the rare variants. So to understand this a bit better, please direct your attention to that diagram. The first line of the diagram shows the presence of variants in the genome as black lines. So imagine that these variants are all rare and they don't occur in the genomes of other subjects. Researchers have very little power, as shown with the yellow stars on the right, to detect these variants. However, researchers can group variants together using genomic annotation information so that they act together as a more powerful unit. So the second line shows an example annotation, which is the gene that the variants are located in, where the genes are represented with gray bars. So grouping variants from the same gene is actually a typical first variant grouping that many researchers perform. And now all these rare variants act together as one unit, and it's more likely researchers will be able to detect an association of their phenotype with variants located in the same gene. Researchers might also perform a filtering step, represented by the third line there, where perhaps they're only looking at variants that are in a promoter region of the gene or genes in a particular pathway, and that can further increase their statistical power. All right, so now I'm going to show some of the tools and features that we have on BioData Catalyst powered by 7Bridges that enable the multiple variant association test. So as a first step, researchers have identified some clinical outcome or phenotype that they're interested in testing. So some examples of that could be iron overload or elevated total serum IgE, high cholesterol. Since researchers want to include as many subjects as possible who have a particular phenotype measured, they might be working with multiple TopMed studies. However, of the 80 or so TopMed studies, they don't use a standardized terminology and measurements for the phenotype data. So this poses a bit of a challenge in that researchers are most likely going to need to perform some phenotype harmonization where they go through a process of renaming, transforming, and processing the measured clinical variables across the studies so that they end up with one comparable variable that's kind of like a one-size-fits-all for all the studies they're going to work with. So how do they do this? Many TopMed researchers are performing the phenotype harmonization in RStudio. So 7Bridges offers both RStudio and Jupyter Lab notebooks built directly into the platform for them to use. This is kind of a first order way though of supporting this part of a rather complicated process. So in the coming months, we're going to be working on developing a customized Docker image for researchers doing the phenotype harmonization to launch, and it will have specific packages that are helpful in this part of the process. Since BioData Catalyst is launching in the coming months or next week, we expect to onboard researchers who already have their own harmonized phenotype data. So then they could use one of several options on the platform to just upload or import this data, like the command line uploader or the desktop uploader. Moving on to getting the VCF data for the subjects of interest, BioData Catalyst hosts the TopMed genomic data in the format that it's hosted on dbGaP. So this means that researchers are getting approval to work with the TopMed studies at the level of consent groups. And these consent groups dictate the data use limitations for study participant data. And there could be multiple consent groups in a particular study. So I'm showing an example here with the Jackson Heart study that has 4 different consent groups. And so the first one dictates that researchers could use this data for all health, medical, and biomedical research, as long as they also have the IRB approval. There are other consent groups that are, for disease-specific research, but still with the IRB approval. And the point I want to make here is that for each of these consent groups, there are multi-sample VCFs for all of the subjects included in that multi-sample VCF group, but then separated out by chromosome. So this means if a researcher is going to be working with data from multiple TopMed studies or multiple consent groups, they've got a handful of multi-sample VCFs at the chromosome level that they're going to be working with. So to find these VCFs on the platform, the researchers can go to our data browser feature, and they can select from the list of hosted TopMed studies shown here. So let's say, for example, they're working with subjects from the Jackson Heart Study here and the Framingham Heart Study, because both of these studies have measured a similar phenotype. So they're checking the boxes next to both of these studies and then saying explore selected. And on the following page here, they can use the visual graph-based query builder to specifically search for the multi-sample VCFs and the consent groups they have access to. So in this case, they've chosen the file entity, and then they're searching over the data type property where they can pick the variant call format. That's the multi-sample VCFs, in contrast to selecting, let's say, like the aligned reads, that's the CRAM files, or the single sample VCFs. They can also pick the consent property to search over, where you can see Jackson Heart Study and Framingham Heart Study are both listed, and they can check the boxes next to the consent groups they have access to. They'd hit this refresh button down here, and they're down here in the file list, and then copy them to their project. All right, so now the researcher has multi-sample VCFs from 2 consent groups. For running the association test, though, they're going to need to merge the VCFs from the different consent groups together, keeping the chromosomes separate. So for this part of the process, Seven Bridges recommends the BCF Tools Merge tool. We host this on the platform in the public apps gallery. This just shows the tool description page here. So it's ready to run and available for all users on the platform. All right, so now to touch on the association tools, there are many different types, and Seven Bridges has primarily focused on providing the Genesis R package association tools, which were developed by researchers at the TopMed Data Coordinating Center for performing a GWAS on TopMed studies and other similar non-homogeneous data sets, because these tools really have a robust ability to estimate and account for population and pedigree structure, which is a key component of the TopMed studies. It's really important when analyzing them to be able to account for that. They have both single variant and multiple variant association tests available. And then just a quick note that the researcher goes through a process of fitting a null model once under the null hypothesis of no genotype effect. Seven Bridges worked with the TopMed Data Coordinating Center to bring the Genesis association pipelines to BioData Catalyst in the common workflow language, which we've touched on in our various presentations in this workshop. So this screenshot shows the workflows in our public apps gallery after the researcher has searched for Genesis. So then they're seeing these 5 different workflows pop up. They've all been optimized for the cloud and set up such that they're stringing together many individual tools into single workflows to make it as easy as possible for users to set up and run, so that, instead of having to run 10 or 15 separate tools, the researchers are going through these 3 major steps listed on the left. They're converting their VCF files to a different file format that's needed for these tools. They're fitting their null model, and then they're running one of the 3 association tests. So they've got single variant, aggregate association, and sliding window. These 2 here are the multiple variant tests. All right, so this is the task setup page for the aggregate association workflow. Just to highlight that users can make use of the default parameters for the workflows set by Seven Bridges, or they can use this interface to easily change these app settings, easily change the parameters without having to go in and change the code. And then the phenotype data and the VCF data that we talked about are serving as inputs for these workflows. And there's one additional input file for the aggregate association test that I want to mention, and that goes back to the variant grouping information that I referred to earlier. So when working with these rare variants, the researchers need to create variant groupings based on that genomic annotation information to increase statistical power. And so researchers might be grouping together variants located in the same gene, treating them all as one unit. The Genesis aggregate association pipeline needs an input file with this variant grouping information. So Seven Bridges spent a lot of time working on building out a service that enables users to query over the annotations for all the top med variants. And we call this service the Annotation Explorer. So this screenshot here is showing the user interface for the Annotation Explorer, where researchers can choose from about 300 different annotations to aggregate and filter over. For example, if you're looking at the filter down here, the users picked 6 different annotations to filter over. And then up here in this table preview, you see that they can see columns for each subject with these 6 different annotations. When they finished going through the process of doing their filtering, doing their aggregating, they can export the results, and that's how they'll get that file in their project that they can use as an input for the aggregate association pipeline. So to end, this just shows the completed task page for the aggregate association pipeline. This is where users would go and be able to access their result files. And with that, I will take questions.
Could you tell us a little more about that variant annotator? How deep does it go? Do users traditionally need to go to other sources, or is that sufficient for their analysis?
Yeah, the idea is that it's including annotations from a number of different databases and annotation pipelines. So it's supposed to be sufficient as kind of the one-stop shop for researchers who need to use that annotation information. And we'll also be building out that feature so that it can be used for post-association testing analysis. So once the researchers identified some variants in their results, they could go back and look at annotations for those variants.
And this is running off the Seven Bridges component, right?
Yes. Yep. Everything I just shared was specifically on BioData Catalyst powered by Seven Bridges.
Thank you.
Like the variance files were separated by chromosome. Is there any advantage to running the analysis on the individual chromosome files, or as opposed to running it all as one large VCF file that has all of that data in one location?
I'm not sure about that. I would have to ask. I kind of just verified with a teammate that it's actually inputting the chromosomes as separate, but I don't know the reasoning behind that.
I think it has to do with some of these top med BCFs are so large, having one BCF that's like a terabyte is really difficult to interact with. So splitting by chromosome can be a little bit easier on the end user, and also for parallelization in the workflows.
Thank you, Beth.
No problem. Great presentation too, Allison.
Thanks.
So I have a question. I'm not sure if you know it or not. how does Seven Bridges actually interact with the Gen 3 data model? Do you guys re-index and it's presented in a different way to end users than the graph structure?
Yeah, I think that's right, because it's being mapped to our file management system, Vapor Store, that Manisha touched on a little yesterday in the back-end presentation.
Okay.
But we are using a lot of the metadata provided by Gen 3 in their manifest for what is searchable to users in the data browser feature. So what's shown in the data browser feature is only open access metadata, so that every user on the platform can search over the exact same things. So it's meant to just be a quick way to get users to like the multi-sample VCFs or the CRAMs. It's not intended to do like what Windmill and Picture are doing with, you know, advanced phenotype search.
Okay.
Yeah.
Thanks.
And Allison, if I can note some of the initial conversations that we're having with the Seven Bridges team specific to BioData Catalyst around being able to eventually ingest a PFB sent from Windmill that will, you know, encompass a lot of the same kind of conversations that have happened with Terra in terms of how is that visualized and adapted within Seven Bridges. And that's just, I think,
Yes.
early days, but it's upcoming work that we have across us, at which point the relevance of that graph model will become much greater for the Seven Bridges team.
Yeah, that's something I didn't have time to touch on fully, but that this process could start with a user on Windmill or on Picture doing a search of the top mid phenotype data to form their cohort before they're on Seven Bridges and working with the VCF files there. Thank you, Garrett. Okay, should I pass it off to Allison Heath?
All right, thank you, Allison. I guess I'm the other Allison. Hi, everybody. Good to be back after my ad hoc introduction of Kids First yesterday. And thanks so much to Allison for giving that really great introduction, kind of making up for the deficit I have of not having great slides. But I think in a lot of ways, the things that were just articulated about TopMed and BioData Catalyst really have the same impact on Kids First. So similarly, we're looking at oftentimes very rare variants. There are lots of cohorts here where there's actually been a lot of whole exome sequencing done and not much found. And so most of the data in Kids First is whole genome sequenced. And also the other kind of unique feature is that most of them are trio-based studies. So we are also getting the sequences of the parents, sometimes even other family members. And so as was just articulated, oftentimes to do some of these GWAS studies, you need a fair bit of power, you need to bring together different cohorts. Kids First, we also have those kind of GWAS types of studies, and it's actually one of the impetuses for why we want to interoperate with BioData Catalyst, because all the hard work we just saw, we don't want to have to redo bringing over Genesis or other things like that, while at the same time we have some unique use cases, especially around the trio-based analysis where you're really looking on a family level, individual patient level. And then I'll talk a little bit about how really we're starting to try to push the boundaries in terms of how does this get translated into clinical care. So, doing the GWAS, doing the landscape, we've discovered a lot, we know a lot. And I think there's a lot of open questions on how do our platforms help really start getting closer to the translational, both research and potentially even clinical care space that we want to be at. So I haven't been on this whole time, and I hope everyone's holding up okay with the virtual setting of this. But I thought, I kind of assumed that there'd be a lot of discussions around Gen 3. There's been a lot of discussions around the Seven Bridges platform. So we've really built on that really strong foundation, really leveraging Gen 3 for the data management, the data cataloging, the authentication authorization, which really sets us up well for interoperability there, as well as the KFDRC platform, which has many of the similar features that you've seen both from Dave and Allison presenting. So what I thought I'll do with my time here is actually to talk about some of the things on the Kids First platform that we've built specifically for our program, but also thinking about how can we leverage that across the different platforms. And then I'll talk a little bit, I think, in terms of the use case. One of the key cohorts that are of interest is something called the Pediatric Cardiac Genomics Consortium. So this is a very large consortium that's been traditionally funded by NHLBI to understand how genomics impact cardiac defects in kids. And so it's an interesting setup, and I kind of wish I had slides about this, but there's been a PCGC DCC, very similar to the TopMed DCC, which has cataloged a lot of this information, done a lot of work with investigators themselves. The data has been in a consortium model, and really with the whole genome sequencing being done both by Kids First and by TopMed, there's starting to be a increasing interest of how does that work come into these platforms. And I think it's really a driving use case where we have some investigators who want to do things like the GWAS that was just shown, while we have other investigators that are looking to do more, phenotypic profiling, clinical outcomes. For example, for pediatric heart defects, one of the major interventions is actually surgery. So how do we collect information about surgery? How do we look at that on a family-based level, et cetera? So that's all kind of a preamble to say, I'm actually just going to go ahead and start showing some of the features of the platform and walking through some of what we've built and how it connects to what you've seen already. This is, again, informal. Feel free to interrupt me or feel free to ping in the chat if I'm going too fast. I know often clicking through these sites can be a little bit of a whirlwind. And so, again, feel free to follow on. Everything I'll be logging in here to should be open to everyone. Although I'll see what we get at the time. We have some back-end tools of how we actually run and operate our platform, and I'm happy to talk about that as well. But kind of from the user researcher's perspective, hopefully everyone can see my screen. It's the main website. And similar to anybody else, you have a portal login. I'm in incognito mode, so Facebook doesn't work. So if Google and OrcID, I'd say that those are the two main things that we see people logging in with. And so anyone can log in. There's no whitelist or anything like that. It's fully open.
Could you make your browser a little bit bigger?
Sure. Is that better?
Yes.
So ORCID is for the Kids First Data portal?
Right, so you can pick any of them. The idea though is that you wanna keep it, although I obviously have an account under every one. So I was using my ORCID ID here. Some of the thoughts around that is that we might be able to track publications, might be able to connect people doing interesting research later on. But the Kids First portal, the login is really for two purposes. One is initially in this space, we were making some of the phenotypic data that originally had been controlled access open on the structural birth defect side. So these are things at the level of HPO terms, et cetera. And we really wanted to have a process where we have a click through, people know that they're using that data in the right way. But really, I think the second part has been, how do you kind of save state for users? So, you're spending a lot of time creating complex queries. You might be creating virtual cohorts. Anyone who's done that knows you can't just select all and expect your analysis to happen. There's often exceptions. There's reasons why you want to include, exclude people. And so really thinking about how do you kind of save that state as you're doing your research. So I logged in with my OrcID because I just want to show you what it looks like when you're brand new. So it's pretty empty here. You have to set up all the linkages to your authorized studies, set up the linkages to Kavatica projects, and then you have a kind of big blanket view of what are some of the top different studies, frequent diagnoses. And again, similarly, since we do have this member concept, people can put in what are they research interested in, and I can talk a little bit about that as well. So I'm going to go to my real workspace, and I'll make it larger as well. So you can see once you have everything set up, you have different kind of saved queries, both over the participant level as well as a file level if you want to do it on the files. and then authorization. So this is really kind of the integration with Gen 3. And you can see that we have Kids First studies here. So we have kind of all the different Kids First studies. And thanks again to kind of explain the consent groups. People have different levels of consent groups. So we have the different data use groups there. Since I'm an admin, I've heard I have one of the largest dbGaP project applications in the system. So, because I do it all through dbGaP. Sorry, was there a question? There's some background noise. Okay. And so when we first started out, we really had the same kind of classic faceted search that I think a lot of folks have seen, where we had the different clinical filters, file filters. The impetus was really to get the data out there and available because of SRA no longer accepting whole genome data. The GDC was not accepting the Kids First cancer data because it was primarily germline. Really, our first mandate was to get it out there. But then really talking to the investigators and researchers, you realize that the faceted search, while kind of nice to initially explore and browse, doesn't really necessarily meet the needs of querying and kind of fine-grained cohort building that they like to do. So I've created this cohort builder. Oops. And the idea being here to kind of help support that iterative process a little bit. So I think because we are in general a little bit of a smaller program, we've actually put a lot of effort into phenotypic harmonization so far. So we have mapped most of the phenotypes to human phenotype ontology. We've mapped most of the diagnosis to Mondo or the NCI thesaurus, kind of depending on the disease space. And we are moving forward with other kinds of harmonization efforts using Uberon for anatomy, and really starting to think about what other kinds of codes and terminology systems can we align some of the other data sets with. So you can see you have like a quick filter up here. So if you were, for example, interested in Ewing sarcoma, you kind of get a quick glance of, we have one with the study, we have the Mondo diagnosis for it, we have NCIT, and then we actually also preserve the source text of the data that's coming into Kids First. Oftentimes, I think along the lines of TopMed and others, the investigators who are involved in that study have put different kinds of notes here for different reasons. So if you're an investigator within the study, you still might want to have access to the original text. Well, if you're someone coming in for the first time, just trying to understand what's out there, having the harmonized fields are often a better entry point. But you can see, this is part of the iterative process. So let's say you're saying, okay, yes, I am interested in Ewing sarcoma as a whole. Okay, great. But if I think about what I just saw, I want to start a new query, and I actually, don't want these kind of ones with these little notes, right? So here's some here. And, right. It's a little bug that we need to fix. So let's say, so the source text, so I have those, and I actually don't want any of those. So I can apply those. So now I have Ewing sarcoma, I have everybody who doesn't have those, and for example, I could add those together. And now I can actually see I have a couple Ewing sarcoma in the brain tumor study, as well as ones here. And now I've kind of created a cohort, you can see, from the original 370 people to one that's being excluded, and for different reasons. And so this is kind of the process that we see people going through. And again, similarly, you might want to save this, right? So I've probably done this wrong, maybe I'm missing something, or maybe, the other way around when you see it is, clinicians come in here, kind of got in here and said, I don't understand why there's 369. I thought I was excluding them. And someone else can come in who's maybe more familiar with advanced query and kind of adjust it and fix it to what the expectations would be. So we really see this not as a singular user often using these queries. It's often being passed back and forth as people are trying to construct things across different studies with people having different kinds of information about what they're looking for. And everything down here is clickable. So, you know, another kind of example use case we often hear about trying to talk about between cancer and structural birth defect studies is, for example, let's say I want everyone with cancer. And again, you can see the studies filtered down, and then you can see the different diagnoses. A certain individual might have multiple diagnoses. For example, low-grade glioma can progress to high-grade glioma. Or if you have a recurrence, sometimes, especially with kids where there's aggressive treatments with leukemias, we're starting to see secondary cancers occur later on in their lives. So, you know, these are some of the things that I think oftentimes when people haven't really looked at data can be interesting and difficult to actually explain sometimes, and kind of how these counts are made up. So these are some of the things that we've been thinking about how do we, as people are exploring and looking at this data, how are they viewing that? And then I think it becomes even more complicated as we start to integrate other data sets. So here we have our very first integration with the NCI Cancer Research Data Commons through the Data Commons framework. And you can start seeing that we have some, you know, target cases with neuroblastoma and acute myeloid leukemia, because we know those disease types are going to overlap with kids first. But you might imagine if we tried to bring in everything in GDC in this view, some of the trade-offs that you might have to do to help people navigate and look at those different data sets. So similarly, like I said, we've mapped all the studies that come through, we've been mapping to HPO. And so even though these are cancer cohorts, they still have different kinds of phenotypes that are showing up. You know, there's different kinds of abnormalities of skulls, there's neurofibromas, epilepsy. You know, if you think about pediatric cancer, like I said the other day, you know, it's often not environmental. There's other predispositions, there's other kind of abnormalities that are happening. And so to think about cross-disease analysis, you could select all there, and now you're filtering down to, okay, here's all of the people in cancer who've had profiles of phenotypes. And then you could actually duplicate this query and ask yourself, okay, of all those phenotypes I see in cancer cohorts, what do I see in the structural birth defect cohorts? So this would give you a result of all of the phenotypes that I saw in cancer. Here are the participants in structural birth defects that have those same phenotypes. And then you could imagine you could start looking at the different genomic data types that are available and think about doing an analysis on some of the shared phenotypes, for example. Similarly, I'll just show you a CAN one that we did before. This was actually a question that we got about, there's a lot of interest on somatic mutations of, do you see somatic mutations in germline as germline mutations in different kinds of disease types? So in the developmental landscape, sometimes if you get a somatic mutation later on in life, you're actually going to have a different impact as if you were born with that. So this is, for example, all the overlapping tumor normal pairs that we have, and then all of the ones that have congenital heart defect phenotypes. And then, you know, the question would be, can you look at the somatic mutations and how much do they overlap with germline mutations that we see in congenital heart defects, for example? So from there, we do have initial kind of entity pages about specific participants. So you can actually start looking at the different kinds of information that they have there, the different kind of clinical information. So again, like I was saying, a lot of times there are very multiple disease types, phenotypes that are happening. And then increasingly, we were really trying to capture the longitudinal bit about it. So how are those changing over time? When do we know about them, et cetera? So you can see that, you know, this patient has quite a few phenotypes. We also mark observed and not observed. So something that's often key is trying to understand what's not observed. Is that just because we didn't look for it? Or is it because someone actually checked and said, no, they don't have an abnormal liver? And that's going to be very important for the downstream analysis as well. And then again, kind of being able to explore and iteratively go back to, if I select that phenotype, I can now see all the different diagnoses that are associated with that. All right, so that's great. I can get my cohorts, I can kind of explore, I can find what the data is. I think the question always is, okay, what can I do with it then? And so I've been talking a little bit about our integration with Target. So on Kids First, we're only hosting the Kids First data. The Target data is hosted by the GDC in the cloud. But because of this integration and then the ability for the Seven Bridges platforms to cross-integrate, we can actually select across both Kids First and Target data sets, push it to KVADICA for analysis. So I'll just show a quick demo there. So again, we have both neuroblastoma in Kids First as well as neuroblastoma in Target. You can see that collectively you have 1,900 participants. You can see that in Kids First, 609 of them are probands, or the kids with the actual cancer. In Target, right, there's only 277, but combined, I get a much larger cohort. Okay. And so you can see from there, I actually want to look at the actual data files, and this is going to take me over to the previous file repository view where I could then narrow down the actual data of interest from this cohort. So you can see that here there's a lot of file filters. There's even some long read data, for example. But let's say I'm interested in just looking at the whole genome data. I want them aligned. And then I can see that I have kind of the CRAM files from Kids First. These are just the index files from GDC, so they're the BAM files. And I can see that I have, you know, about 1,600 participants there. So I'm just going to do this to kind of show you that this actually works. I could just export all of them, but I can start with the Target data, for example, and then here I can click Analyze in KVADICA. So I can, again, this is calling out to the KVADICA APIs, and I can create a new project here. I can save that, and then I can copy my 14 files. I'm also open to suggestions about verbs of how we want to think about what we're doing here. Copy is not really the best verb because there's no copying of data. We've tried to analyze, we've tried push. I feel like at least having some of the verbiage right on what it really means to go between these portals, these cloud workspaces, might help users throughout this as well. And so you can see in here that we have, within my new test project, the new Target files. And then if I switch from Target to Kids First, again, I could do this all as once. I'm just kind of belaboring the point a little bit that I can then, in KVADICA, also push my Kids First files. I refresh this. I then have Kids First files, Target files on the same workspace. I can bring in my pipeline, run it across all that data, and get my analysis results here in KVADICA. And this is really done by an integration to the backend with the Cancer Genomics Cloud. And I think the hope is that to make all of this much more bidirectional, so people can work where they find the most utility. Any questions about that? Anything that I could answer as I was kind of going through there? So if not, the other thing I want to talk about is that we've been increasingly thinking about how do we integrate this with other visualization platforms. Again, trying not to reinvent the wheel, but really think about what's out there that's existing that people are already using. So for example, we've been thinking about with the piloting with some of the brain tumor data, the ability to go between this and something that was set up by CHOP a while ago, PEDC BioPortal. So you can see for some of these cancer patients, I have a link here. And this takes me to an instance of CBioPortal, which contains both the public information from CBio as well as any of the somatic data that's coming out of Kids First. So again, our purview is not really in the tumor data while it's being generated. And so really we want to be able to reuse, repurpose other tools that are out there. And really what we've been thinking about is some of the visualizations around germline trio based data. But you know, initially a lot of people, this is the space they really want to come into where they see the visualization of the different data, the different annotations, being able to know, you know, what do they mean? And looking at the different data here. So that's our kind of first foray into that. I will say as part of hopefully the new Common Fund Data ecosystem, we've also been talking to the Links group. And so this is a large project to do profiling across cell lines on drug perturbations. So this is a lot of transcriptome data and how do we visualize and think about, you know, now that we have the specific patients, the landscape, we understand some of the mutations that are happening. How does that really impact with the different drug screening that's going on? So these are some of the ideas that we're thinking about in terms of how do we make this data more available, more accessible. Okay.
Hey, Allison, a quick question there. What was the, as you left the Kids First portal and launched into the other one, what was the key that was used to create that URL to go to, I can't quite read it.
Yes, so
the BioPortal. But in other words, how did you come to the agreement on what key to use?
So with how to, yeah, so because these are Kids First IDs, basically in PEDC BioPortal, you can use whatever ID you want. And so we've been just using the same one across the two platforms.
So the downstream portal had imported your data with your IDs.
Exactly.
Gotcha. Thank you.
I think that's an important point. We've run into sometimes when we've done integrations and things won't accept like underscores or like little things like that. Or oftentimes in pediatrics, you know, as you might imagine, a lot of these kids are pretty sick and they get IDs in multiple different places. So for Target, for example, there's actually a Children's Oncology Group ID. And so now we have mappings between Kids First IDs, COG IDs, et cetera. And I think it's actually probably a problem for offline, but you know, a lot of times we actually can genomically figure out that people are the same when we're doing these analyses, and how do we handle that? Mainly because you need to remove duplicates and things like that when you're doing the different kind of statistics, especially when you're looking at rare variants. Okay, yeah, so that's PEDC BioPortal. So I'm gonna take off my Kids First hat for a little bit, and I want to talk a little bit about what I was saying about how do we take this data and bring it to care. So what I'm going to show next is really more on the CHOP research side and kind of what we're starting to do with some of these platforms. And I just want to separate that out a little bit from the DRC hat that I normally wear. And so again, I was saying, okay, this is great. I can take this virtual cohort, I can push it into something like KFDRC. But what we're really starting to see is as people are generating genomic data locally, they want to bring their own patient and start asking, how does this patient compare to the landscape that's in these different platforms? And so we've been initially piloting in KFDRC, again, not as part of Kids First, but rather showing examples of how people can do this, is the idea of I have a new patient, I've created a pipeline on KFDRC that will take in their data, run it through the Kids First pipelines, and actually generate an HTML report. And so we've actually been using this platform. You know, I think a lot of times people think about it as for the computational biologists, the really, you know, people creating these large scale workflows, but actually starting to think about the end result of that. Actually, you can have a collaborative space that people can log into, look at together, and actually use as part of the clinical part. So this is an initial patient report. This is just kind of some test data, but really starting to bring in resources. So this is, for example, are you interested in bringing in links along with the Kids First data? And then I have my initial patient, and I can start asking myself, what's genomically similar? So I have all these pipelines on KFDRC, they're run in multiple places, and they get knit together in this report, and I can start seeing, this particular patient with this triangle here, how does it relate to what we're seeing across different diagnoses? And for example, this is a high-grade glioma. These are where the high-grade gliomas are normally clustered. This is actually clustering with different kinds of gangliomas. And you might ask yourself, why is that? What variants are there? How might I adjust the treatments for it? So thinking about, not just using these platforms for the large-scale analysis, reharmonization that we can do, but how do we really empower individual investigators to do kind of workflows like this? So I just kind of wanted to put that out there in terms of, I think in a lot of ways, this is going to be so important for interoperability because I want to bring in not just the data from Kids First, but other places. And while I'm showing a cancer report here, a lot of the structural birth defects often have metabolite kind of interventions, making decisions on surgical interventions, things like that, that will also be informed by this genomic data. And so, yeah, I think that brings me back to what we've been trying to do with PCGC, where we have the whole genome data in Kids First has been processed. It's actually released in live. It's actually one of the most well phenotyped cohorts. So I think it's also a very good example for how we might do phenotyping. And then really thinking about how do we cross leverage the BDC platform that Allison so nicely presented. So I'm going to do a quick thing that hopefully works, where I am going to disconnect everything that I have connected that makes this look very cool in a demo and show you exactly as a new user just what I have to do to make all this work, right? So now if I were to go back and try to do that same thing, where I as in KFDRC. So tell me I've not connected my KFDRC account. Please follow the instructions. To register for KFDRC account. So this is even like if I'm a very brand new user, I'm going to have to register for a KFDRC account. To go to the developer dashboard, I'm going to have to copy and paste the token here. Once I do that, I then have to go to my settings, log in to the framework services, the NCI, CRDC, etc. So I'm not going to belabor this too much and go through all the steps, but when we first started talking about doing the BioData Catalyst integration, it was like, oh, great, we've already done NCI. We'll just add a third box here. And then you start realizing that it's kind of creating this combinatorial issue for users. And while I think a lot of times it feels like people say single sign-on and stuff is not that key to interoperability, it's amazing how many people just get stuck thinking about like all the different things that they need to log into. So that's part of why I think we've been pushing on some of those aspects as well.
That's really helpful, Allison. All right, so I think that's most of what I had. Are there any questions? Or I could also, if people are interested, I could show you some of our internal tools for ingesting data and releasing data. I was curious, are there ongoing sequencing efforts that are going to lead to new data sets being deposited in the near future in the data portal?
Yes.
I guess I'm just less familiar.
This is Valerie. Sorry.
There is so many.
This is Valerie Clyde. Yeah, so we're still actively sequencing, and we've actually only released 9 out of like 30 something projects so far, and we'll be sequencing for even 1 more year.
Cool.
At least.
And so I guess I will show. So one of our challenges has been because of how the sequencing mechanism is funded, really investigators just get funding for the sequencing. There's no money for phenotypic harmonization. Some of the groups like PCGC have a DCC set up, but a lot of these diseases is just, investigator has been collecting biosamples as they've been a clinician researcher over the years. And so we've had to take in clinical data and stuff from multiple different places, REDCap dumps, spreadsheets, PDFs, emails. And so we started creating this data tracker because, as Valerie said, there's actually quite a few projects coming in. So, thanks to actually, Valerie and others at the Kids First Program Office, we've really been very clear about the embargo times. So data gets sequenced, it gets made available to the investigator, and then within 6 months, we've actually been pretty good about getting into the dbGaP process. The dbGaP process takes a little bit of time, but then as soon as that gets released to the public, we can flip a switch and make the data available. But tracking and managing all those projects as they're coming in, we tried some various project management tools, but ultimately we ended up kind of starting to build up our own application. So this is, as Valerie said, all the different studies that are coming in for Kids First that we're tracking. And you can see that we have information about the study. We have abilities to upload the different files that are coming in, versioning of this because it changes over time as IDs and stuff get mapped. And then we started kind of adding some features, actually similar to BioData in a lot of ways, to be able to tag, to be able to sort and go through these different files. As part of our process on the Kids First side, we have to say we have enough information to release the data to the investigator because as part of their proposal, they did commit to providing certain clinical data elements. And so, we bring that in, we do different kinds of ingestion processes, and then we also create KFDRC projects for them to not only deliver the data that's coming from the sequencing centers, but also kind of another, value add of what we're doing is doing pipelines and stuff that are aware of the families as well as aware of some of the types of studies that we expect people to do. So we run another level of pipelines to create harmonized data there. And so we have the different KFDRC projects tracked here. We also have the ability, and we've been piloting cloud credit projects. So anything under there is also being tracked in here. Kind of logs of everything that was done. You can see, you can actually probably talk to Chris Friedman, who's on the line, who actually knows this app way better than I do right now to really work with investigators there. And then really looking at integrating some of what we've been doing with the release coordination system to actually manage all these studies coming in. So I'll just show that real quick because that's the last little piece. Kind of the back-end system is that we really recognize that we would have to do this pretty quickly, and that if we really wanted to interact with multiple systems, that we couldn't be responsible for all the different ETLs or different ways that people wanted to manage, but rather send out notifications, have people set up what we call task services that report back to us and say, yes, I got the data, it looks good, or no, we have an issue. So this is how, when we're ready to do a release, you can actually plan a release, select the different studies that you're interested in. We have the concept of minor releases, or just a new study or new data, or a major release. If we change the data model, we have other issues like that. And then you can see that there's multiple task services that are registered. So in Portal, we have the 7 Bridges task service. We also do some snapshotting and reporting as well. And so, for example, if we want to test or something, we can enable or disable them. And then you can actually, start a release. I'm not going to go through the whole process, but you add release notes and other things that all get stored in here. And so that's how we have been able to manage the release process, and especially when we first ramped up. I think we were doing releases, every other week or so, and it's been a really great kind of lightweight method to be able to notify system to system that new data is available and have it picked up and then reported back to us, were they able to pick it up successfully or not. And I think this API is open. You do have to be authenticated to actually tell us and write back to us. But I think it's an interesting thought of how do we send notices and how do we make new data available, because to your point, there is definitely more data coming in. This is definitely, we view Kids First as kind of research and process. So we're very aggressive about getting data out there, even if it's provisional data. I think I'm out of time, but I'm happy to take any more questions offline, or I also want to kind of do a shout out to Chris Friedman and David Higgins, who is onboarding during this pandemic time, but is going to be our new scientific community manager. And Chris Friedman's been our data support analyst, who's really been doing a lot of work getting the data flowing into the system. So thank you.
Thank you, Allison. Next up is the ISB CGC/CRDC use case. I'm not entirely sure who's leading that, so feel free to grab the screen.
I'll be doing that. Can you hear me?
We can hear you loud and clear.
Okay, excellent.